Hi, I am Cheyenne Lupton, and this is my first video for Exam 3 in Genetics 311. This will be on the Meselson and Stahl DNA replication experiment. This experiment was done in 1958, which was right after the structure of DNA was discovered. The new goal after this was to learn how DNA was replicated. There were three theories: conservative, dispersive, and semi-conservative replication. Conservative replication is when a whole new duplicate DNA is made. The result is one old DNA and one new DNA. When it replicates again, there is just one old DNA left and three new DNA pieces. Dispersive is DNA replication that is a hybrid of both old and new DNA in both strands. Both strands are put together and it stays that way no matter how many times it is replicated. In semi-conservative replication, each individual strand acts as a template for a completely new strand. Each DNA has one old and one new strand. After replicating twice, there are two hybrid DNA pieces and two completely new DNA. So which one is right? Meselson and Stahl set out to figure out which replication was correct. Meselson and Stahl used E. coli bacteria for their experiment. The bacteria was grown in a heavy isotope of nitrogen, also known as N15. They let the bacteria replicate many times. After the DNA with N15 was grown, the researchers transferred the E. coli DNA to a lighter isotope, nitrogen-14, or N14. They then let the DNA replicate, and then the samples were analyzed using density centrifugation. Density centrifugation separates DNA bands by spinning them with a different chemical. The heavier strands sink to the bottom, while the lighter bands stay on top, showing the different DNA chemicals present based on their weight. After the first generation was replicated with just the nitrogen-15, a single band was shown because there was only one chemical. The next generation, which contained both N14 and N15, also produced one band. However, this band was higher than the first band, indicating that N14 and N15 had mixed together and created a hybrid. This rules out conservative DNA replication. When the generation after that was analyzed, it produced two bands. One band was in the same place as the previous generation, but the next band was above that one, which indicates that it only contains N14. The result proves that DNA replicates semi-conservatively. Overall, Meselson and Stahl proved that E. coli bacteria replicates semi-conservatively. It was later proved that this replication not only applies to bacteria, but to nearly all forms of life.